Spatial biology is something that was always around. Okay, there is a new omics every day.
How far are we from one instrument?
I'm gonna be very provocative here. Why do we need one instrument? There are companies that they say, oh, we can do both. Then you speak with the user and they say, no, they can't.
Hey guys, it's the first video that we're filming dedicated to our guests in life sciences, in multiomics and spatial biology, and some other exciting topics. And today we have our first guest, Giovanni Russo. And today we'll be talking about spatial biology. Let's start. Let's start. Hello, Giovanni. Really big pleasure to have you here. Thank you for traveling all the way here to Amsterdam. Today we would be speaking about spatial biology, why it is so exciting to work in this field, and what is this field actually about, because I feel like it's quite new. 10 years ago I don't think people talked about it that much, at least in Europe. And we will start with introduction. Tell us who you are, introduce yourself.
First of all, thank you for inviting me. I am Giovanni, but everybody calls me Gio, shorter. And I am Italian and I live now in Germany since more than 8 years. I am a former scientist, so I did my PhD in neuroscience, working a lot with imaging. And then I moved to industry because I didn't like science, or actually academia so much. And I decided to move to industry to still be part of the world of science, but in a different way. I worked in a lot of markets, imaging, omics, spatial biology, and always in customer-facing role. I started from the basic field service engineer, building instruments and training customer, then moving towards application specialist and now sales. And what I like about my job is I still speak and live with science, with scientists, but I don't have the pressure of writing grants, writing papers, or looking for money all the time. And also that I am exposed to a lot of different projects, a lot of different kinds of science, a lot of new technology that are just arriving in the market. For me, science is not only a job, it's also a passion. So I'm also like to keep myself updated about what's going on in the world of science, and especially regarding new technology.
Okay, that's fantastic. We can maybe dig a little bit more into the world of spatial biology and what is so special about spatial biology.
Spatial biology is something that was always around. We just changed the name multiple times. Because if we look at how people are looking at samples, we just put a piece of tissue under a microscope and we try to look what is inside. What changed is that the technology got better, which means now instead of visualizing one single target, we can label hundreds of thousands of targets or a full genome or a full proteome, which means we can have a lot more information than before. This also implies that the way we are dealing with this data is changing. It's becoming more and more interesting and more complicated, because previously we were analyzing the data by hand. So you were having a human eye, looking at the sample and saying, this is a cancer cell, this is not a cancer cell. Now we have software that can do that. Previously we were having a microscope that were manual. Now we have microscopes that are automatized. And previously you have to change buffer or solution by hand, now that we have liquid handlers. So spatial biology is the combination of an old technique with a lot of new things coming from automation, software, and better detectors and better probes for labeling stuff, for labeling structures.
For me, as a person who comes from the academia, I'm curious to know your opinion, because you're mentioning that spatial biology is a field that is evolving very rapidly, and you have automation on top, data processing on top, and the companies are in the race for whoever can provide better technology for that. And here I'm curious because you've been on the different sides of the industry as an engineer, as application scientist, right, to some extent as a salesperson. Academia has enormous pressure to publish papers, and they are also very often the first user of the new instrument and new platform. And do you sometimes see the resistance from the scientists when there's a new instrument on the market to buy it, because they may feel that, well, if we will automatize everything, what are we there for?
There are some elements to consider here. First of all, the people. Some people are very open to innovation. Some people are not very open to innovation. In my experience, Europe is very conservative. US is much more open to innovation. But once a technology is well established, is then Europe where you have more opportunity of success. So if you have a product that is somehow still in development, then it's easier maybe to have somebody in the US to be happy to be an early adopter. But if you want to be really successful, then you need to come to this side of the pond, so you need to come to Europe and get in the big KOL. If you look at the market of spatial biology, for example, you will see that the main player are American companies. They all come more or less from the same place, which is California. Then they started the development, they co-developed with scientists, and then they came to Europe. And then in Europe, they engage with KOL. Yes, scientists there are under a lot of pressure for publishing. And usually to publish, you also need to have something that is innovative. But companies are under another kind of pressure, which is the pressure of creating something that can provide this level of novelty. Level of novelty that can also be supported by a certain level of stability. Because if you have a machine that doesn't work, then you are not publishing. And let's keep this in mind. It's not complicated to build a machine, okay? I know people that they can run spatial biology experiment on their own machine, and it works very well. But that machine is designed for one purpose. That is the purpose of that lab. So the obstacle for a company is to produce something that can be flexible, so fulfilling multiple purposes, still providing good data. And this is why it's important to have early adopters. Usually you should look for very ambitious postdocs or young PIs, but then you need to get somehow the big player on board, which helps you establishing yourself into the market. The innovation part, in principle, from a hardware perspective, there is not so much to innovate. The tools, the machines that are around are pretty good, pretty strong. It's just a matter of combining things together. The difference comes, for example, from the chemistry. So which kind of probes are you using? How these probes are binding the structure of interest, which relates strongly to the quality of what you're seeing. And also, what matters is the software, because now we generate terabytes of data. So analyzing this data manually, either you recruit an army of PhD students, or you will spend years just to get an image for a single well. So software for data analysis is, in my opinion, where companies are fighting more, because you need a good software that is producing reliable data in a good way. It's also nice to see how there is currently a switch between wet lab and computational lab. I am old school. So when I was doing my PhD, my postdoc, I was doing data analysis. So it means I was doing my experiment, putting myself on a chair, working for hours with a lot of coffee, analyzing data. Now we have software that can do this almost automatically with a little bit of supervision. And especially when we are looking at images, cell segmentation, shoal analysis, or all these kind of tools are very important because these are what people are looking at. Because the beauty of spatial biology is that we are adding a layer of information to standard genomics or standard proteomics. So when you sequence something, you know that you have, for example, a certain genes, but you don't know where this gene is and how certain genes are spatially correlated to each other, or how certain RNAs are spatially correlated to each other, or protein, can give an indication of a pathological condition versus a physiological condition. And this is very difficult to be done just manually. So this is why software is very important in this new kind of technology.
Do you think that, well, with a world of AI and technology evolving every day, do we have potentially the tools to understand this data, or are we limited by the computational capacity and the only appearance of, let's say, quantum computers will enable the answers for us? Or is it just about, instead of producing new platforms, we need to focus more on the quality and structured data analysis and about labeling everything properly, and kind of this holistic approach of all the scientists agree on how do we label it and how do we interpret it?
I like to say holistic approach and scientists agreeing on doing something, which in my experience is very difficult to get two scientists to agree on something. I partially disagree because we have already way more powerful tools than before. If we think about the new generation of a GPU that can process terabytes of data in parallel, that already allow us to extract this information much faster. Problem is that when you look at the data, then you need to give an interpretation. And that's where the human part enters and plays the role. Now, machine learning, artificial intelligence, of course, will speed up this process. There are already some companies looking into that direction, so analyzing data and providing hypothesis or verifying hypothesis, which helps to reduce. And this is becoming, for example, more and more relevant in cancer biology. There are some companies where you have tools where the software is analyzing the image and giving options about, for example, the grade of the tumor, so level 4, level 3, or metastatic or non-metastatic, infiltrative, not infiltrative. And that's already happening. Are these tools perfect? No, but they will never be, because at the end of the day, it's the quality of the data. We will get to perfection. No, we will get to excellent level of approximation, but this requires that we need more initiative of people coming together, sharing data, with a desire of knowledge and not a desire of being the best, and sharing this on a base where people are looking into how we can better analyze this data to get better prediction. At the end, these tools are developed to improve the quality of our life, to have early detection of some pathologies or better diagnosis. So if we keep this in mind, and if this becomes the main reason, not only for the user, but also for the companies, then I can see in the next years data analysis become much faster, interpretation as well, and having better diagnostic tools. And this is already happening today. It's just that, yeah, maybe when we will have even more powerful computer, the quantum computers, for example, all the countries are working on this. this will become even much better.
And what's your take on the, this bias interpretation? Because when you take the images that were labeled by scientists make mistakes. It's a human thing. And as we know with, this famous experiment about training language models, where you have in one language, the feminine gender or masculine gender, and the translation usually goes like, she cooks or he is smart, this kind of things. I'm talking about those mistakes, because it's propagated through the centuries in literature. That's why we have this bias existing. Do you see that this is potentially a bottleneck in science when we analyze the images that were labeled by doctors, for example, who made the mistakes, and now those mistakes potentially would propagate into the science, and what are the mechanisms?
Well, the mechanism is there, so the bias will always be there. Even the best trained artificial intelligence or machine learning tool will do mistakes, because it's normal. What is possible to do in order to minimize is to combine evidences. If we look at the market, many companies that are in the genomics space, sequencing space, are entering the spatial biology market. And now they want to enter the proteomics market, and then the lipidomics market, and then I don't know what omics market. Okay, there is a new omics every day. Multi-omics. Yeah, let's call it multi-omics. There is a new omics every day. And that's nice, because if we combine multiple evidences, then we can have a better interpretation of the data. If we have a monodimensional point of view, then this brings to bias. An example, this is an historical example. In the past, one single doctor was looking at the glass light and giving the diagnosis. They changed it. They added another doctor. And that massively reduced the number of mistakes. Say 2 sets of eyes are better than 1. Now, let's think about having not only more sets of eyes, but more evidence combined together. That will reduce the amount of mistakes. Mistakes that are always done in good faith. But if we have this opportunity of combining multiple data, extracting multiple points of view from the same sample, then in my opinion, it's much easier to get a better diagnosis or a better interpretation of what is really in that sample.
That's great insight, and I was always curious because I feel like indeed the companies are emerging and they're trying to have a portfolio of the instruments that covers as many dimensions as possible. And in your opinion, how far are we from one instrument that is able to do, let's say, genomics, proteomics, transcriptomics, and some lipidomics? Because, they're kind of...
I'm going to be very provocative here. Why do we need one instrument?
It saves the cost for the lab. Instead of buying 3 instruments, finding the place, organizing logistics, having different teams training you, you have one instrument that has different functions. And actually, do we need it? Like, do people in the lab and doctors, do they need it?
When you buy a coffee machine, you look for the kind of coffee you prefer. I'm Italian, I look for something that is very good in doing espresso. Okay? So I don't care, for example, if that coffee machine can do a cappuccino. Scientists will look for the technology which fits better his or her requirements. If we look at a facility, it's very difficult that, in my opinion, that a facility is going to acquire a machine that does everything. Because it's almost impossible that you have a company that is capable of building a machine that is good at everything. So, can we get a machine that is the perfect machine for spatial biology, spatial transcriptomics, spatial proteomics, spatial lipidomics, whatever omics? Maybe, potentially, not shortly. but we can get a combination of machines. Omics is an example, genomics is an example. We have a provider for short read and long read. There are companies that they say, oh, we can do both. Then you speak with the user and they say, no, they can't. They are not really, they are better for long reads than short reads, other way around. Also, would you trust a single machine for everything? Because this is like going back to one set of eyes. It's not better to have a combination of machines. You know, I know a lot of people that they run their samples on 2 different machines. And then they compare the results and say, why you're running the same sample on 2 different machines? Why? And the answer is simple. Well, because it's normal that there is a difference, but if what I think it's there, it's really in there, I must see it in both. So this is why I think we don't need a machine that fix all from an hardware perspective. It's okay to have multiple providers, multiple machines. Every machine top range, excellent for a specific application, proteomics, transcriptomics, genomics. Then maybe it will be nice to have quality check. So tools that allow to assess the quality of whatever we do and whatever is coming out from a machine, which is something that, especially in the field of spatial biology, is still missing, and it's a little bit of a limitation. So no, I don't think we need a one machine. I think we need, let's say, level of quality that every machine must reach. And then, well, I'm very happy to say, let's get multiple machines together so we can analyze the data in a different way to see if then we all reach to the same place.
So reflecting on this, in the field of biotech, when you have a new technologies entering markets, there is still some time before they go into the clinical space and before they're really tested and used for specific diagnosis or for predicting of how certain drug will, interact and give a certain effect. And I was wondering if, well, first of all, the fact that not all the machines are producing the results at the same quality level is a bottleneck, or is that what are the bottlenecks for the spatial technology to enter the clinical space, and which technology you think is the closest, or maybe already there, that is used?
Definitely the lack of a quality check is the problem that is limiting the access of spatial biology into pharma and clinical application. I had a lot of conversation with the people working in pharma and pathology, and they all say, show me the protein, show me the immunohistochemistry. And you're like, why the immunohistochemistry? We can do spatial biology. Yes, but the immunohistochemistry has 20-25 years of history. When you will have 25 years of evidence proving that, maybe you will be right. Is it a conservative approach? Yes. Is it wrong? No. Let's think about somebody who gets a biopsy and then is waiting for a diagnosis of a cancer. Do you think that the patient wants the cool technology or wants the safe technology? The patient wants the safe technology. Do you think that the doctor that is going to deliver the information wants to play the game of being cool and using the new technology with a chance of saying, yeah, you are perfectly healthy, and the patient is not healthy and is going to die? No, they don't want that. So the point here is we need to get the new technology at the same level of the old technology, re-establishing, first of all, verifying some of the main points already existing to say, we are as good as, and now showing we are better. And this is what, in my opinion, sometimes is not happening, because spatial biology is mostly used still in academia. And there are some nice, beautiful initiatives now to translate this into pathology. There are some good players in Europe, for example, doing this. There is Dennis Shapiro in Heidelberg is doing an amazing job in doing this, or Jean-Philippe Malm in Heidelberg is a great advocate for QC in this kind of, in spatial biology. And he's also, he's a great guy, he's a very outspoken person, very honest, which I love about him, even when he's not delivering good news. But at the end, they are going towards that direction. Then when we will get the same level of safety, I think this will happen. But this implies, again, establishing quality check and establishing that level of confidence in saying we are at least as good as what we were using before. And this not only to somehow fulfill that desire of safety from the customers, but also that fundamental requirements that when we are giving a diagnosis to someone, that diagnosis must be right. Because you want to know if you have cancer or not. So we need to be sure that that diagnosis is correct. And at the moment, I still don't see this as being fully proved or fully tested. So it should also be a little bit more of play with the pathology department and pharma company in order to make this happen fast. But on the other side, they have to look at the legal consequences of wrongdoing. So it's going to take time, but it's getting definitely better.
Gio, let's discuss platforms in spatial biology. Which technologies do you think are the most promising?
Nice question, complicated question. So there are two main technologies. One technology is sequencing-based or amplification-based. The other one is imaging-based. They are both excellent. It strongly depends from what you want to do. For example, for small panels, I think imaging-based technology are still the best. You have excellent resolution, very low false positive, so amazing. But if the panel gets too big, then you have something called optical crowding, so the quality of the data gets worse and worse. And this means that if you're looking at a small panel or well-defined RNAs, that works great. Same for proteins. But sometimes maybe you want to look at the full genome or the full proteome. So in this case, amplification or sequencing-based technique are better because you have the full overview. It's a little bit correlated to what I was saying before. Can we have a machine that does both good? No. Can we combine the two machines into a single machine? Potentially. But maybe a good platform can purchase both machines and then running the two experiments at a different point in time. So maybe running on amplification sequencing machine base for looking at what are the most promising RNA or protein, and then going to another kind of machine to dig deeper into this kind of technology. It's the same, for example, in microscopy. You can start with bright field imaging, then move into confocal, then move into super resolution in order to get deeper insight into the sample.
How do you see the field of spatial changing in the next 5 years?
I think that the future will see, again, most of these small companies becoming part of something bigger and the technology be integrated. Then I hope that they will work a little bit more on establishing the QC, and also that they will somehow also looking at outside of the conventional field, which is cancer biology. Everybody's doing cancer biology, super interesting. But for example, there is neuroscience, there is immunology, there is also all that section that is related to organoids, which is growing as a market, and is also related to a clear request of the FDA, for example, about reducing the number of human experiments of animals. So that's where I see the field moving. So small company becoming part of bigger companies, technology that are gonna go more towards high-plex for, let's say, widespread research and discovery, and low-plex for validation. Multiomics, almost everybody now is doing already RNA and protein. Probably will be RNA, protein, DNA, and lipids in the future. So then you can even add metabolomics, but this is gonna be controversial. And then let's say the final part will be the establishment of when a QC will come out, so people will know what is the minimal quality to be delivered to the customer.
And personally, how do you see yourself contributing to this development?
Well, myself, I hope that I will have the chance to work again in this kind of field, the field of spatial biology or in the field of proteomics. And I hope that I will have the chance to having a customer-facing role again, where I can collect the feedback from the customers, from the users, and bring it back to the company, so they can be implemented into the next generation of machine, so we can produce something better, something that works faster, more efficiently, and also allow us to go a little bit beyond what we know now. So let's say to the next step. This is what I hope I will do in the future.
Let's go back to the roots. You studied neuroscience.
Yes.
While spatial biology mainly known in the field of cancer research.
Yeah.
How was your transition? And it's not only that you studied neuroscience, your first job was as a field service engineer, but recently you've been doing sales. Seems like you've done quite a few transitions. How was it for you? And
well, yeah, I'm a neuroscientist. And I have a PhD. Now, when people ask me what having a PhD means for me, I always reply that I was taught about how to learn, how to being capable of looking at information, understanding them, and using them, which means for me it was pretty easy to move from neuroscience to cancer biology, immunology, oncology, but even plant biology, because some parts are always the same. Biology is less complicated than what people think. It's so, for me, it's always so surprising to find out how many genes we have in common with a plant and how many processes are the same. Then, of course, there are differences. But if you know the basic, then you can grasp the more complicated level. Of course, you need to study. You need to be willing to ask questions. Then, regarding my first job as field service engineer, well, I was coming out of academia, and I knew I didn't know business. And personally, I thought, what can I do? Now, I like building stuff and repairing stuff, something that I do as a hobby. So my fiance is always, oh, we need to change the tire of the cars. I do it. Oh, I need to change the lamp. I do it. So I always have I have a lot of tools at home to repair things. So I was not afraid of working with tools. And a little bit as in science, I was thinking, if I want to understand something, I need to know how this something works and is done. So field service is that kind of job. And it's also a job where you have a lot of meeting with customers. You spend a lot of time with customers. So a salesperson spends 1 hour at dinner with a customer. A field service engineer, depending on the machine, spends up to 2 weeks with a customer.
Yes.
So 2 weeks with a customer, you become friends. You know how many beers you drink in 2 weeks? So, but it also teaches you the deep nature of the machine. And once you understand the technicality, then you can go higher. It's also a very nice introduction to business. Because within field service, there is a little bit of sales, there is a little bit of application. Then you can go to the next step. That can be application, where you are technical, more related to the biological or chemical consequences of what the machine is doing. And then there is sales, where you need to be good at presenting the machine, but you also need to be good at answering the question about the weakness of the machine, because no machine is perfect. So it's very easy to say, oh, my machine is the best. Yeah, but then the real struggle is when the customer, and nowadays customers are spending a lot of time on internet looking at reviews, is asking you, yeah, but I read that the lab of Professor ABC in the University XY is complaining about the fact that your machine cannot do this. How do you reply? And that's where, if you are coming from service, if you're coming from application, you know how to answer. Because also then the point is, how important is this weakness for the customer? Is this a deal breaker or not? If it is a deal breaker, then you need to fix it. If it's not a deal breaker, still you need to fix it. But then the approach change. And honestly, and here I know I'm going to be provocative, I am very confused recently because I see a lot of people coming out of university and going directly for very high positions. And then, poor them, I have to say, they fail. Because university doesn't teach you business. A PhD doesn't teach you anything about business. It doesn't teach you about how to deal with people at all. You are excellent at dealing with animals, probably, but not.
That's the best case scenario. Usually, it's just
Python
and you.
Or you're very good. You're very good at keeping yourself alive, which is a little bit different than keeping a customer happy. So then, people enter jobs without knowing what they are doing. Sometimes they are immediately tossed in the middle of the field without proper training. And that's the problem when then you have companies struggling and people struggling. Sometimes you have also people that are into jobs without really knowing the jobs. I met product management, they don't even know how the product looks like. How you can manage something that you don't know. And too often, I think companies forget one very important point. Customer, they don't care. If I am entering a lab or an office and I am wearing the t-shirt with the brand of the company I'm working for, they have a problem, they are expecting me to be capable to help them. Now, I'm not saying a sales must be capable of fixing a machine, but a sales must be capable to point the customers towards the right direction for obtaining a solution. Or the point is also that, and there the solution is not, as I heard from someone, well, just wear plain clothes. No, that's not the solution, that's stupid. Point is, we need to get back to the point that we work with people. People that they have needs and they have problems. We are selling solutions that are supposed to alleviate the problems, not create more problems. And behind a machine that doesn't work or doesn't deliver, there is a PhD or a postdoc that is struggling. PhD maybe is not getting the data to finalize the thesis. A postdoc is not getting the data to finalize the paper for the grant application. And then there is on top the professor that maybe is in tenure track and they need that paper. So whenever we are doing this, we need to be able to provide a solution. If we don't know what we are doing, if we have people that are not capable to really understand and explain what they are selling, then we create unhappy customers and we set up people for failure, which explains why there are companies that are burning sales faster than cigarettes. So this is why I think we maybe, and I know this is very provocative, we need to go back to the point where we are sure that everybody knows training is provided. Even a salesperson should be capable to use the machine a bit. Also because you get more credibility in front of the customer. And then seriously, how can you sell something that you don't know? So that's why I decided to start from what is seen as the lower job that you can have, which is absolutely wrong, because field service people are fundamental in the success of every company, and they're also most of the time the one customers remembers. Then, of course, I wanted to challenge myself with different kind of jobs. But this is the reason why I started from, let's call it the bottom, which is not the bottom.
I don't share the opinion that it's the bottom. I think it's one of the most important jobs and the most important hires that the company usually makes, because those people indeed at the front of the customer, they set the tone. If you're not able to, not on the level of the field service engineer, on the global level, because it's not everything that field service engineer can fix, right? Sometimes the machine is just not meant to work in this way. But the field service engineer should be able to spot it at the right moment and to deliver this information to the company, and the company should take action. So it's a very important role. And
when you go to a lab, it's like which numbers do you have on your fridge? Personally, I have the number of the plumber all the time, the number of the garage where we repair the cars, and the number of the doctors. These three, they have in common that they fix problems. A lab, same. They have the number of the field service engineer, the name of the field service engineer. When they get to solve the problem, that is hardware, then they have the number of the field application specialist when they want maybe some advice about science. And then there is the number of the sales when they want to complain about something. That's the point. That's how it works. And everybody has a job. Everybody has a role. In my opinion, the job of the sales is to help the customer to get the best with the money that they have, and to work in collaboration with the field service engineer and the field application specialist, so the customer is constantly happy. Then the job of the field service is that the machine must run as best as possible, and the job of the field application specialist is that the customer is using that machine best of his conditions. But these three people, they are a unit. They work together. Because again, the customer, even if the customer knows that you are sales, is going to look at you. If you are going visiting the customer and there is a problem, they're going to report you. So if you are capable of, for example, extracting a log file, customer will say, oh, good, you know, or I remember one time I entered a lab and the customer was like, the machine doesn't work. Why? Just a machine was something stupid. In principle, was a cable unplug. Yes. You just plug the cable again, and was like, it works. Great. And now that's exactly the point. So not only the customer got a good service, but the customer got, wow, they care. No, Geo cares. The company cares. Because the sales is trained in a way that can take good care of me. And that's what's built reputation, which then becomes getting introduction to other customers that brings to revenue. But if the purpose is, we need just to generate revenue with nothing behind, then you have nothing at the end.
Yeah, especially if you look at the big established companies, if you look how they make money, it's not most of the time from selling capex, it's from selling everything that goes into this package, which is services, which is consumables, etc., etc. So at one point, you need to deliver the services and support, and not push for big machine that would, stay there and work or not work.
Without mentioning, one of the company I was working for was making more money with service than with sales. And actually, there were more opportunity to give a discount on buying a machine than on service contract and consumables. And the level of attention in training the sales engineer, training the field service engineer, was very, very strong. Because the engineers must be all capable, professional. They had to deliver continuously. There was a lot of pressure. But that company has a very excellent reputation in terms of field service. And they do a lot of money with service contract, consumables, because if you're selling capital equipment, you will not sell capital equipment every year, especially when the price is high. Nobody's gonna buy. It's very difficult to find somebody capable to buy a machine that costs 500, 400,000 euro every year. At one point, that market will die. So you need to have a market that is done by consumables, service, training, and that's where you see we're gonna get at the end of the race. Pure capital equipment companies, they don't last long. They are generally acquired. Smart company, they try to diversify, selling consumable, selling service, selling training, producing new machines. The market is showing what's going on right now. There is a company opening every day, a new company coming out every day, and a company failing every day. Again, even there, before opening a company, the main question is, do I have a product somebody wants to buy? And even if somebody wants to buy it, what I am gonna do next? Because at one point after I sold 100 machine, what I do? Cool, I sold 100 machine. Nobody's buying anymore. There are recent examples of company that they even sold 1,000 machine and then they got acquired because the market dry up.
Yeah. Nobody to sell to.
Exactly.
Gio, what's your favorite thing about the field of spatial biology?
Well, the first one will be the images. They're amazing. But in reality is that I think spatial biology is bringing together the best of multiple technology. We have high quality imaging, plus we have high quality chemistry, high quality software. So we are bringing out the best of multiple worlds, and it allows to see things that were unthinkable before. 15 years ago, thinking about visualizing RNA, DNA, protein in the same sample, people were looking at you and say, oh, that's crazy. Now we can do it, and we can do big panels. And this is making the life of clinicians and academics better. also is challenging. So we have, for example, recently there was a beautiful paper about something called STAMP, where they're using spatial biology for sequencing. So that is the measure of how innovative this technology is, how much value can be added in what we always did, and also a value of how we have a long path to go to get better. Because the technology is great, definitely needs improvement, which is also the things that I like the most. Some technology are so well established that it's impossible to be innovative, or it's very difficult. Spatial biology, no. Spatial biology is still young. So as such, it's like dealing with, I think now a teenager, so let's hope it stays in a good mood, but there is the opportunity for growing and going to the next step. This is what I really like. And again, my home office is a picture of images of spatial biology from different platforms and providers, because they are super amazing.
Well, thank you very much for this conversation. It's been a pleasure to have this chat, and yeah, stay tuned for our next episodes.